the important issue of dietary fiber and the way it affects cancer now. And with me is Jenny Paredes from the City of Hope Medical Center in Duarte, California. But Jenny, you're at the San Diego meeting of the American Society of Hematology right away, all very exciting. And you're talking about increased fiber intake, and you apparently have better overall survival and lower incidence of graft versus host disease in patients who get an allogeneic stem cell transplant. Now, can you tell me what led up to this work? Because there's a bit of history on this, isn't there?
It is. So first of all, thank you so much for your invitation. And I'm from here, sharing from San Diego, from ASH. The starting point of this project is about a decade of investigation of the relationship of the intestinal microbiome and the outcomes in bone marrow transplantation. And so my lab, the lab of Marcel van der Brink, who's now the president of City of Hope, he had already found that when you have a more diverse intestinal microbiome and you have a higher abundance of bacteria that ferment fiber, they're called butyrate producers, you have actually bettered overall survival to GVHD. So that was already established. So when I came in as a microbiologist, and I have always been interested in nutrition oncology, I proposed the idea if we could use fiber as a dietary intervention to increase butyrate producers, to increase fiber fermentation, to increase the diversity of the microbiome, and therefore increase overall survival. So that's my starting point.
You have been able to do a study. In your study, you've got two parts to it because you've got human patients and a good sample of them too, and you've also got an animal model that proves the principle. Tell me what you did in the study.
That's right. So our strategy includes, as you mentioned, human and animal studies. So we're very proud to have a cohort of 173 patients from the halogenic stem cell transplantation, and we follow the dietary intake of these patients every single day, every single meal, every single drink. So we have a data set of close to 39,000 meals that we recorded for 5 years. We also corrected every single meal for the portions that were eaten because it's very important to have accuracy of the portions that we're eating. And in addition to that, we collected stool samples every 2 days to monitor the changes in the microbiome due to diet. And so we have close to 2,000 fecal samples that are paired with this dietary data set. So we had a very rich data set from these patients.
And what data did you get out of it then?
So we have amazing data, and I would like to enhance the fact that I focus on fiber, but we have data also about sugars, proteins, fat consumption, and so there, we're just starting to understand a lot of dietary patterns. So from these intense follow-ups that we did, we were able to correlate survival after up to 24 months of these patients and correlate these with the grams of fiber that they ate every single day, as well as the composition of the intestinal microbiome.
Right, and you also investigated graft versus host disease. What came out of it in the statistics there?
That is correct. So, in addition to addressing overall survival, which of course is the most important clinical outcome there is, we also focus on GVHD risk. And we found that patients that had higher fiber intake not only had lower cumulative incidence of GVHD in general, but they have a specifically lower incidence of lower GI-GVHD, which is one of the most severe versions of GVHD. So we were very excited to see these results that not only benefits all GVHD patients, but specifically prevents lower GI GVHD.
Right, now you've got an increase in overall survival. I believe you have a P level of 0.04, which means it's statistically significant. How big a difference did it make in terms of months and years for the patients in overall
So
survival?
in terms of survival, I would like to, we have to be cautious with the numbers, but there is a gap of 10% differences between the high fiber cohort and the low fiber cohort, and this gap of 10% is persistent throughout the 24 months. So we're comfortable reporting 10% increase. up to 24 months.
You did well, of course, getting data because humans typically, a typical sample, would not include a very wide spectrum of dietary fiber, would it? Or did it?
No, especially in the population in the United States, the average population already has a lower fiber intake, and I think it's important to acknowledge that patients that go through bone marrow transplantation, which is a very involved treatment that includes radiation, chemotherapy, the conditioning regimen is quite rigorous. So that actually makes patients naturally lose their appetite. So encouraging patients and designing meals that they can easily have fiber intake is something that we invite all clinicians to improve the strategies.
And to give more scientific muscle to your study, you added a model arm and found out what happens in mice because with the mice you could vastly change the amount of fiber going in, couldn't you?
Exactly. So we also wanted to perfectly like understand the mechanisms of fiber, and there are always many questions that could be addressed, but I think that we got very interesting answers. So number one, as you mentioned, I designed three fiber concentrations. One, the control, which is the recommendations that they always feed mice in every animal model, which is 6%. And then I increased this concentration by a fold to 12% to create a rich fiber diet. And then I had a negative control that was zero fiber. And then so mice with a rich fiber diet, which is 12%, had better survival. They had more abundance of beneficial commensals, beneficial bacteria, such as fermenters of fiber. They have lowered opportunistic pathogens, which is a big problem in the clinic. As we know, bone marrow transplantation patients are subjected to opportunistic pathogens and infections. And in addition to that, we understood by single cell sequencing, so we're trying to keep, state of the art. So we performed single cell sequencing and we understood that part of the benefits is that fiber actually preserves the integrity of the colon, the integrity of the lower GI, and also that the inflammation that is produced by T cells is reduced in a rich fiber diet. So we were very excited about our mouse results as well.
And of course, I've got to ask you about the sort of fiber. I don't suppose you're able to look at this, but it's not just cellulose or things that just cause roughage. There's a whole range of fibers from vegetable products, isn't there?
So for the mouse models, actually, we were very rigorous and reductionist, and we kept it only cellulose because cellulose, we mammals like mice and humans don't have the enzymatic machinery to break down cellulose. So we were confident that all our results are dependent on the microbiome. That was our intention. For the humans, however, we did the classification of the seven most common fibers. So we have cellulose, hemicellulose, pectin, residual starches, etc. And we actually are putting our publication together where we have analyses of specifically different fiber types in our patients' data. So we're also very excited to, in the near future, be sharing those as well.
Right, now, coming out of this, importantly, what would you say are the recommendations for cancer doctors and perhaps for the population coming out of your findings?
Thank you for your question. I would love to highlight exactly what you asked. I think that the beauty of dietary interventions is that the patient can also be a great contributor to their treatment, right? We're very excited to see that our data suggests that patients that had a higher fiber consumption prior to transplantation tend to maintain higher levels of fiber consumption during treatment. So that I would like to applaud the patients for being conscientious of these choices. But to your question about the clinicians, that is correct. Bone marrow transplantation is a challenging setting to introduce raw foods because, of course, they can come, even if you wash them really well, it's a potential risk factor for microbiome infections. So raw foods, no fruits that can be peeled, those are limitations that we need to acknowledge, and they're there for a reason. That being said, I invite clinicians to redesign the clinical menus that are provided at the institutions and to include more diverse sources of fiber. So, for example, an avocado has 5 grams of fiber, and people love avocado in breakfast, in lunch, and dinner. So it could be included, kind of like be sneaking in the three different meals throughout the day. We also invite to think of other sources of fiber that are not commonly addressed, like quinoa, like seaweed, which are great sources of fiber. And again, they can be cooked, they can be blended, they can be thrown in a soup. We're also looking at lyophilized kale and lyophilized spinach as a powder, as a form of supplement that is not artificial, but it will give opportunities for clinicians and hospitals to integrate fiber when patients are not able to tolerate full meals. So we can be very creative with the strategies. And I would last also mention that 10 grams increase of fiber consumption made a difference in our data. And 10 grams, as I was mentioning, it could be accomplished by an extra avocado and an extra small cup of vegetable soup a day. So I think that these are the strategies that are feasible. And so I invite the clinical community to be more creative.
I'm getting hungry already. Some of the fibers you've mentioned are some of the tastiest foods. But what would you sum this up as for doctors? What's the big take-home message? Would you say?
I think the take home message is that, of course, we want to be cautious of patients that might not tolerate a high fiber diet right away during symptoms like IBD like symptoms. And I think that, of course, this is personalized medicine and each case is different. But I think the take home message is to strongly rethink the menus that are being offered to the patients. Bone marrow transplantation patients are on average hospitalized for 40 days. So pre-transplantation, post-transplantation is an average of 30 to 40 days. So this is a beautiful window for us to play our part and make sure that our patients are getting their fiber in their diet.
Well, Jenny Paredes, thank you very much. You're from the City of Hope National Medical Center in Duarte, California. Right now, you're in San Diego, not too far away from there.
And
And
it's
I have
very
the
sunny and
privilege.
beautiful.
It's lovely to connect with you, Jenny. And thank you very much indeed. I'm Peter Goodwin for the Audio Journal of Oncology.
Thank you so much for the invitation.